Good morning, dear students. This is your teacher Neelam Helawat, and we are doing the chapter 8, that is the cell, the unit of life. Dear students, so far we have talked about the various organelles that are present in the eukaryotic cells. Like we have talked about the cell membrane, the cell wall, the endomembrane system. And we know that the endomembrane system is comprised of the endoplasmic reticulum, Golgi apparatus, lysosomes, and the vacuoles. Now, in this particular video, we'll be discussing the further cell organelles, that is the mitochondria. So, we'll complete this list and we will start with mitochondria first. I hope you have heard the name mitochondria. Can you see in the picture? Mitochondrion. So mitochondrion is basically a singular word, while mitochondria, it's a plural word. Okay. Now this mitochondria, everyone, it's a cell organelle that becomes more clearly visible when you stain them. So you can easily observe the mitochondria in a slide under the microscope. Okay. The number, the shape and the size of the mitochondria, it varies from one cell to another, or rather I should say it depends upon the physiological activity of the cell. Means if the cell is more active, it will contain more mitochondria. The number of mitochondria will be more. And if the cell is less active, then the number of mitochondria will be less. Now this particular organelle is a sausage shaped or a cylindrical. As you can see in the picture also, it is a cylindrical shaped. It has a diameter of about 0.2 to 1 micrometer and the length is approximately 1 to 4.1 micrometer. So it is more lengthier in comparison to the breadth. Now basically if we focus on the mitochondrion, dear students, it's a double membrane structure. As you can see here, this is the outer membrane and this is the inner membrane. So mitochondria is surrounded by the two membranes, the outer and the inner membrane, and the space between these two membranes. Okay. Can you see this? That is called the intermembrane space. So there is an outer membrane, there is an inner membrane, and the space between the two membranes is called the intermembrane space. Okay everyone, what do we call this space? We call it as intermembrane space. And this inner membrane, okay, the inner membrane, as you can see here, this inner membrane is filled with a dense homogeneous substance which is called the matrix. Okay, so the inner membrane is filled with the homogeneous substance which we call it as the matrix. So your mitochondria basically, students, you've understood, it is surrounded by two membranes. The outer membrane is smooth one. The inner membrane is thrown in number of folds, as you can see, and these folds, they are referred to as cristae. There is a space between these two membranes. That space is called intermembrane space, and the inner membrane is filled with a substance which is called the matrix. Okay everyone? So what are the cristae? The folds that are present in the inner membrane. Okay. Now basically the folds that are present in the inner membrane, that is the cristae, they increase the surface area. Okay, they increase the surface area so that large number of enzymes they can work at the same time. अब जो अंदर वाली मेम्ब्रेन है उसके अंदर बहुत सारे फोल्ड्स मौजूद हैं जिससे कि उसका सरफेस एरिया बढ़ जाता है और एक बार में बहुत ज़्यादा एंजाइम्स उस पर काम कर सकते हैं. Now as I told you, dear students, that it is surrounded with two membranes. The two membranes have their own enzymes associated with them. Means both the membranes, they have there are specific enzymes which are associated with these membranes. Okay. Now in the matrix, I told you the space inside the inner membrane is called the matrix. Now in the matrix there is a circular DNA. Can you see this purple color DNA? Can you see the purple strands of DNA? Yes. So in the matrix there is a circular DNA present. RNA molecules are present, ribosomes are present, but these ribosomes, they are of 70S. Okay, although the eukaryotes, they contain 80S ribosome, but in the mitochondria there is 70S ribosomes are present. Okay, so we can say this is a cell organelle which has its own DNA, which has its own RNA molecules, it has own ribosomes. This means it can produce its own proteins as well. And mitochondria, they divide by using a process which is called a fission. Okay, means one mitochondria can easily divide into two. So this process is called as fission. Now we are talking about the mitochondria. Can you guess what is the significance of mitochondria, or what are the functions of mitochondria? Dear students, the mitochondria is very, very important organelle. It is called the powerhouse of the cell. I hope you heard this name. Can you tell me, have you heard this name? Powerhouse of the cell. Yes. So tell me, what do you mean by the powerhouse of the cell? Yes, very good. It is called the powerhouse of the cell because it stores ATP. So basically it is the site where aerobic respiration occurs. Okay, it is the site, it is the organ in the cell where the aerobic respiration will occur, and during the aerobic respiration we know that there is 36 ATPs that are produced. So those 36 ATPs, they will be stored inside the mitochondria, and whenever the cell needs the ATP or requires the energy for doing the various biochemical activities, so mitochondria will be supplying the energy. Okay, so mitochondria will supply the energy needed by the cell for its various functions, and that is the reason that it is called the powerhouse of the cell. The next organelle that we have is plastids. Have you heard this name, plastids? Yes. Yes, Ishika, right. Plastids basically, they are only present in plants. They are absent in case of animals. So plastids are the cell organelles which are only present in the plants. And they are also present in few euglenoids. Now this plastid cell organelle, as you can see in the picture. Can you see in the picture? Chloroplast. Have you heard the chloroplast? So chloroplast is one of the plastids. Now they are large in size and they can easily be observed under the microscope. Plastid जो है, इनका size जो है काफ़ी होता है, and we can easily observe them under the microscope. And they contain specific pigments. इनके अंदर जो है specific pigments मौजूद होते हैं, and those specific pigments that gives them specific colors. तो जो pigment उनके अंदर मौजूद होता है उसी के हिसाब से इनका color होता है. Okay. So based on the type of pigment which is present inside these plastids, we can classify the plastids into three types. One is a chloroplast. As you can see here, can you tell me the color of the chloroplast? Yes, it's green color. Very good. So chloroplast. Second is chromoplast. So they will have red, orange, yellow color. Leucoplast, they are colorless. As you can see in the picture also, they are colorless. So they do not have any color. So chloroplast, they are green in color. Chromoplast, they have color other than green like yellow, orange, red. So the pigments which are present in them, that gives them their specific color. Now let's talk about the chloroplast first. So chloroplast, these are the plastids, they contain chlorophyll. Okay, they contain chlorophyll and they also contain some carotenoid pigments. इनके अंदर जो है carotenoid के pigments भी present है. And both these pigments, they help in trapping the sunlight, which is essential for photosynthesis. तो ये दोनों pigments मिलकर plants में sunlight को capture करते हैं, जिससे कि photosynthesis यानी photosynthesis वो process है जिससे plants अपना खाना बनाते हैं. So basically chloroplasts, they help in photosynthesis and they capture the sunlight. The second is chromoplast. Chromoplast means colored plastids. So as I told you, they contain the pigments like carotene, xanthophylls, and they have a color that is yellow, orange or red color. The third category that we have is a leucoplast, which are colorless plastids, and they vary in shape and size, and they store nutrients. So they, in totality, they function as they are the plastids that store nutrients. Amyloplasts, they store starch, and the example is potato. Eleoplasts, they store the oil and the fats. And the last that we have is the eleuroplast, which stores the proteins. So we can say that plastids basically can be divided into three types. This is your chromoplast, chloroplast and leucoplast. Okay. Then your chromoplast, they may be green or they may be red, yellow or orange. Then the leucoplast, they can be further classified into three types: amyloplasts, eleoplasts and eleuroplast. So basically, or in totality, or in the nutshell, the leucoplast, they store starch, oils, fats and proteins. Now let's discuss the chloroplast in detail. We know, dear students, that basically the chloroplast, which contains chlorophyll. Now this chloroplast or the chlorophyll, it is present in the leaves in the special cells, and those specials you can see they are mesophile cells. So as you can see in the picture, these are mesophile cells. Okay, can you see the picture? These cells all are mesophile cells. So your chlorophyll is present in the mesophile cells, and they are lens shaped, they may be spherical, they may be oval or they may be ribbon like organelles. So their shape also varies and their number also varies. So number may vary from 1 to 20 to 40 per cell in the mesophile. Okay, so हमारा जो number है chlorophyll का, वो ग्रीन एल्गी में 20 से 40 पर सेल होता है, जबकि क्लैमाइडोमोनास में इट इज ओनली 1 पर सेल. Now this chloroplast is also consisting of double membrane structure. Like as we have in case of mitochondria, the chloroplast also has double membrane structure. One is the outer membrane, another is the inner membrane. The outer inner membrane, they both are smooth membranes. And the space which is present inside the inner membrane is called as stroma. तो अगर आप outer inner membrane को हटाते हैं, कट करते हैं, तो जो space आपको नज़र आता है inner membrane के अंदर, वो कहलाता है stroma. Now this inside the stroma, there is an organized flattened membranous sacs. And those sacs, they appear as a pile of coins. तो आपको देखने से ऐसा लगता है जैसे बहुत सारे coins का pile arranged है. Okay. So there is a pile of coins which are arranged. This pile of coins is called as thylakoids. Okay, this pile of sacs is called as thylakoids. And it appears as a pile of coins, right? Now one of them is called as granum. देखिए आपकी ये इतनी सारी layers इसके अंदर ऐसे मौजूद हैं. सो वन लेयर इज कॉल्ड एज ग्रेनम. जो एक लेयर आपको नजर आती है वो कहलाती है ग्रेनम. एंड दिस क्लोरोफिल इज प्रेजेंट इन द थाइलाकॉइड्स. जो क्लोरोफिल पिगमेंट है हमारा वो थाइलाकॉइड्स के अंदर मौजूद होता है. नाउ थाइलाकॉइड्स दे आर कनेक्टेड विद ईच अदर. Okay, they are connected with each other by the membranous tubules, and those membranous tubules they are referred to as stroma laminae. They are referred to as stroma laminae. Okay, so double membrane structure, and when you remove the double membrane structure there is a space, which space is called stroma. Inside the stroma there is a pile of coins. Those pile of coins is called a thylakoid. Now one layer in the thylakoid is called as granum. And the membrane of the thylakoid, it encloses a space. That space is called as lumen. Okay, that space is called as lumen. So we can talk about that. See, you can see they are connected with each other. Okay, now in the stroma, okay, this is your plastid. This is the double membrane structure. When you open it, there is sacs of granum. That sacs of granum is called as thylakoid. Now inside the stroma there is circular DNA molecules are present. So in addition to the chlorophyll, circular DNA is present, ribosomes are present, enzymes are present, and they bring about the synthesis of carbohydrates and proteins. So in addition to the mitochondria, okay, in addition to mitochondria, plastids also have their own DNA, they also have their own ribosomes, and they also have their own enzymes. So mitochondria and plastid, these are the two cell organelles which have their own DNA, ribosomes and enzymes. The only difference is that the ribosomes present in the chloroplast, they are smaller than the cytoplasmic ribosomes. यहाँ जो आपको ribosomes नज़र आते हैं या present हैं वो cytoplasmic ribosome से उनका size छोटा है. In the cytoplasm there is 80S ribosomes are present, while in case of mitochondria as well as in case of plastids the 70S ribosomes are present. So now let's talk about the ribosomes. What are ribosomes? Now ribosomes, they were initially discovered by a scientist named George Pallet, and he observed the ribosomes in 1953. So he was the first scientist who could observe the ribosomes as dense particles. Okay, he observed the ribosomes as dense particles under the microscope. Now these dense particles, they were made up of ribonucleic acid, that is RNA, and the proteins. So they existed as dense particles that were made up of RNA and proteins, and they were not surrounded by any membrane. So basically we should remember this, ribosomes, they are non-membranous. All your organelles, they are surrounded by a membrane, but ribosomes, they are not surrounded by a membrane. Okay, they are not surrounded by a membrane. So if somebody asks you, can you name a cell organelle which is not surrounded by a membrane? So that is your ribosome. Now let's compare the ribosomes that are present in the prokaryotic and eukaryotic organisms. Now in case of prokaryotic ribosome, everyone, see, prokaryotic ribosome is made up of two units, the 30S unit and the 50S subunit. So the bigger unit is 50S and the smaller unit is 30S subunit. Now this 30 sub S unit is further made up of 16S RNA. जो आपकी 30S subunit है ये भी 16S RNA से बनी होती है, बल्कि जो आपकी 50S subunit है वो आपकी दो तरीके के RNA से बनी है. One is 5S RNA and second is 23S RNA. And in the nutshell, in the totality, the 30S subunit combines with 50S unit and they form a 70S functional ribosome. So दोनों मिलकर 70S functional ribosomes बनाते हैं प्रोकैरियोटिक में, सेल्स में. Now in terms of eukaryotic ribosome is made up of two subunits. The smaller subunit is 40S and the bigger unit is 60S. The 40S unit is made up of 18S RNA, while the 60S unit is made up of 28S RNA, 5.8S RNA and 5S RNA. So 3 types of RNA clubbed together to form the bigger unit, that is 60S RNA. And when these two subunits they combine with each other, they form the 80S RNA. So in general we can say that in case of prokaryotic organisms, prokaryotic cells, the ribosome is 70S, while in eukaryotes the ribosome is 80S. When this ribosome it combines with the mRNA, okay, this is your mRNA. So when the smaller unit combines with the larger unit plus the mRNA, 3 things: smaller unit of ribosome, bigger unit of ribosome plus the mRNA, then in that case we call it as polysome. Okay, what do we call it as? We call it as polysome. So polysome is made up of the small unit, the bigger unit and the mRNA. Right everyone? I hope you understood the concept of ribosome. And then the protein synthesis will start. So first of all, the smaller unit will combine bigger unit. Then both of them they will combine, or you can say ribosome will combine with mRNA and they will form a polysome, and then the production of protein will start. Okay, I think this much is clear everyone. Okay. Now as you can see here, just let's see the eukaryotic ribosome. Smallest unit is 40S, the biggest unit is 60S. Okay, and this 60S is made up of 20S RNA, 5.8S RNA and 5S RNA. So in totality 45 proteins. Here 50S RNA, in total 30 proteins. And then both of them they will join together to form the ribosome, the functional ribosome, which is your 80S. The next component that we will talk about is the cytoskeleton. Cyto means, cytoplasm. So cytoskeleton, what is cytoskeleton? Cytoskeleton is a network, okay, it is a network of filamentous and proteinous structures which are present in the cytoplasm. साइटोस्केलेटन जो है वो एक नेटवर्क है जो फिलामेंट से मिलकर बना है, और ये जो फिलामेंट्स हैं ये इनके अंदर प्रोटीन्स प्रेजेंट है. Now as you can see here, this is actin filament. Now actin filament will form microtubules, and then they will form the intermediate filament, and then they will form the complete structure which is called a cytoskeleton. Now this particular part, the cytoskeleton component, it is involved or it provides mechanical support. ये सेल को मैकेनिकल सपोर्ट प्रोवाइड करता है, मोटिलिटी प्रोवाइड करता है, मीन्स इट हेल्प्स इन मोशन, एंड इन एडिशन टू दिस इट ऑल्सो मेंटेन्स द शेप ऑफ द सेल. सो ये सेल की शेप भी मेंटेन करता है, जिससे कि प्रेशर की वजह से उस सेल की शेप चेंज ना हो. ओके, coming to the next component, that is your 8th component, cilia and flagella. I hope you have heard this word, cilia or flagella. Cilia, the singular word for this is cilium. Flagella, the singular word is flagellum. So whenever there is a single flagella present, we call it as flagellum is present. Now these cilia and flagellum, they are the hair-like outgrowths of the cell membrane. They are hair-like outgrowths that are coming from the cell membrane. Cilia basically they are small in shape and they work like oars. Do you know what are oars, dear students? चप्पू. देखिए जब आप बोट में बैठे होते हैं तो आप चप्पू चलाते हैं. So that oars they help in movement of the boat. Similarly, these are very small structures which are present around the cell, and they work like oars, and with the help of them the cell is able to move, or it is able to propel itself in the surrounding medium. And cilia, they will always be more in number में ज़्यादा होते हैं, small होते हैं size में. Flagella, they are basically bigger in size but less in number. Okay, they are small, they are big in size and they are less in number. So both these particles like cilia and flagella, both these cell organelles, they are covered by a plasma membrane. As you can see here, both of them, they are surrounded by a plasma membrane. And in the center of the plasma membrane, there is the core. They consist of a core, and that core is called as exoneme. Okay, center में core present होती है, उस core को हम कहते हैं exoneme. Now this exoneme consists of a number of microtubules, and these microtubules, they are running parallel to each other, as you can see here, right? This is a longitudinal section of the exoneme. So in the center you will find a structure which is called the exoneme, and this exoneme, as you can see in this, is consist of parallel microtubules which are arranged, and there are 9 pair of doublets. अगर इन माइक्रोट्यूब्यूल्स को हम स्टडी करते हैं तो बेसिकली देयर विल बी 9 पेयर ऑफ रेडियली अरेंज्ड माइक्रोट्यूब्यूल्स, एंड इन द सेंटर देयर विल बी अ पेयर ऑफ माइक्रोट्यूब्यूल्स. सो देयर इज एन अरेंजमेंट ऑफ 9+2. Okay, so in cilia and flagella there is an arrangement of 9+2. Let's have a better understanding of this in the next slide. So this is your exoneme. This is a structure of the exoneme. ये आपका complete structure है exoneme का. इसके सेंटर में देखिए 2 central tubules, central microtubules present है. So we can call it as, there is a pair of centrally located, okay, central microtubules. Now on the periphery, on the outer side, there is 9 peripheral microtubules. बाहर की तरफ अगर आप देखेंगे तो 9 peripheral tubules हैं, और वो भी doublets में है. doublets means pair में है. So 9 pairs of peripheral microtubules are arranged. Now the central microtubules, they are separated from the peripheral microtubules by a sheath. Okay, so one sheath, a central sheath, separates the central tubules from the peripheral tubules. Okay, and the peripheral tubules, they connect themselves with the central sheath. जो आपकी peripheral tubules हैं वो central sheath से connected हैं, and that connection is called as radial spoke. उस connection को हम कहते हैं radial spoke. Okay, now this arrangement we call it as 9+2 arrangement. So this is a 9+2 arrangement of microtubules, and this complete structure is called as exoneme. ये जो पूरा structure है इसको हम exoneme कहते हैं. तो आपकी cilia और flagella जो है वो exoneme से बने होते हैं, means 9+2 array system उनके अंदर present होता है. I hope this, I think this is very clear. You've understood the radial spoke, you've understood the central sheath, and now see. Now peripheral tubules, as you see here, peripheral tubules, they are connected with the central sheath by radial spokes, but they are also connected with each other. ये structure आपको नज़र आ रहा है, जो आपकी peripheral tubules हैं, microtubules हैं, ये आपस में भी connected होती हैं, और उनके बीच का जो आपस में connection है उसको हम कहते हैं inter-doublet bridge. Okay, so your peripheral doublets, they are interconnected with each other by the linkers, and those linkers are called as inter-doublet bridge, while they are also connected with the central sheath by the radial spoke. तो आपकी जो cilia and flagella हैं, दोनों organisms जो है, sorry, दोनों parts जो हैं, दोनों cell organelles हैं, the cilia and flagella, both of them, they emerge from, or they arise from the centriole-like structure, which is called as a basal body. तो cilia और flagella दोनों जो है वो basal body से arise होते हैं, basal body से form होते हैं. I hope everyone has understood. Or somebody wants to share something? Okay, is this much clear? Okay. Now let's come to the next part, that we have the centrosomes and the centrioles. Centrosomes and the centrioles. Now these are the last cell organelles which are observed in case of eukaryotic organisms. Centrosome is an organelle that contains two non-membranous cylindrical structures called centrioles. So अगर हम टोटल in जनरल बात करें तो ये आपका सेंट्रोजोम का स्ट्रक्चर है. सेंट्रोजोम के अंदर दो सेंट्रियोल्स प्रेजेंट है. Rather we can say two centrioles make a centrosome. So if we understand the structure of centriole, we can easily understand the functioning of centrosome. Okay, and these centrioles, they are surrounded by a pericentriolar material. ये जो centrioles हैं ये आपस में interconnected हैं और इनके बाहर एक layer है जिसे हम कहते हैं pericentriolar material. Now both these centrioles, they are arranged perpendicular to each other. They are not lying parallel to each other, rather they are arranged perpendicular to each other. And thus it appears, it gives them a shape of a cartwheel. तो इस तरीके का shape आता है जैसे एक cartwheel है. Now these centrioles, they are made up of tubulin protein. जो आपके centrioles हैं ये आपके tubulin protein से बने होते हैं. And how many fibrils or how many fibers will be there? What is the total number of fibrils that will make up a centriole? 9. So centrioles, they are held perpendicular to each other and they are made up of 9 evenly spaced fibrils, and these fibrils, they are made up of tubulin, and each of the peripheral tubulin fiber is in the triplet form. Okay, it is in the triplet form. Let's see it. If we can see the structure, let me draw the structure for you. Now suppose as I told you, they have a structure like this. So triplet is there, 1 2 3. So 1 2 3. So they are arranged in triplets. Like in case of cilia, there are doublets are there. आपने देखा cilia के केस में जो है doublets हैं. अब यहाँ पर एक और ऐड कर दीजिए, then it will become a triplet. So in case of centrioles, triplet is present. So 3 triplets, they will make 1 fibril, and the adjacent triplets, they are also joined with each other. So 2 centrioles, they will make a centrosome. Centrosome is surrounded by a material which is called a pericentriolar material, and these centrioles, they are held perpendicular to each other. They are made up, their fibrils are made up of special protein which is called as tubulin. The central part of this centriole, see this is a centriole, these two together are called centrosome. The central part of the tubule of this centriole is called as hub. The central part of the centriole is called as hub. Now this hub is also connected with the tubules. Okay, this is also connected with the tubules by radial spikes. And the centrioles also form a basal body, as the basal body is present in case of cilia and flagella. Okay, now from the centriole, the spindle fibers will arise. जो हमारे centrioles हैं उसके अंदर से spindle fibers arise होंगे, and these spindle fibers, they give rise to spindle apparatus which is formed during the cell division of the animal cells. So हम कह सकते हैं कि centrosome और centriole दोनों का function होता है that they both of them they help in the cell division process, or they help in separating the chromosome or the chromatids, and the separated chromosomes or chromatids then move to the opposite poles. Thus leads to a distribution of DNA in both the cells. So they play a very crucial role in the cell division of animal cells, and they are only present in the animals. They are not present in the plant cells. So with this, we finish the various cell organelles that are present in the eukaryotic organisms. Now dear students, your homework for today is that you will be making notes of this topic. You will read the page numbers 135 to 138, especially focusing on the words which are highlighted with black. If there is any doubt, anything that you want to say, you want to understand, or there is any query that you need to ask me, you can send your queries personally to me. Okay? I hope I am clear, everyone. Okay, so that's all for today. Bye everyone.